Hello everyone. In the last class, we have discussed about the basics of genes, genomes and chromosome, and we have also seen the molecular structure of gene. But that is not fully, we just learned the basics. So we will continue the some more concepts in the genes, genomes and chromosomes. So here you can see the title, that is monocistronic and polycistronic mRNAs. So we know that mRNA is also called as messenger RNA. The nucleotide sequence which is present in the mRNA will determine the amino acid sequence in the polypeptide chain, because the ribosomes which are present in the endoplasmic reticulum in eukaryotes or the free ribosomes which are present in the prokaryotes will determine what the amino acid sequence should be present in the polypeptide chain, that is newly synthesizing polypeptide chain, only based on the mRNA nucleotide sequence. Okay, we know about mRNA. But what is the monocistronic and polycistronic mRNAs? A cistron is a genetic unit encoding a single polypeptide chain. So cistron means here a genetic unit. Genetic unit here is nothing but the nucleotide sequence. Okay, so a cistron is a genetic unit which encodes a single polypeptide chain. Cistron means it can encode a single polypeptide chain. So mono means single. So monocistronic means the genetic unit or the mRNA molecule which can encode the single polypeptide chain is called monocistronic. And polycistronic means a single mRNA which can encode the more than one polypeptide chain is called polycistronic mRNAs. Okay, if you see, most of the bacterial mRNAs are polycistronic. Example, lacoperan, that every graduate, at least at undergraduation stage, we will know about the lacoperan. So to metabolize the lactose, the bacteria will synthesize three types of enzymes. That three types of enzymes are nothing but the proteins. So they should only come from the mRNA. That means the amino acid sequence which is present in the enzymes also should be encoded by the only mRNA. Okay, so that single mRNA will contain the three cistrons. So if it is having more than one cistron, we can call it as polycistronic unit. So the lacoperan mRNA contain three cistrons which can encode for the three enzymes. Okay, so each cistron containing one ribosomal binding site within the polycistronic mRNA. If you see the polycistronic mRNA, each cistron having the ribosomal binding site. So the ribosome will bind to first enzyme cistron, first enzyme sequence which is first nucleotide sequence of cistron which encode the first enzyme. So it will synthesize. After finishing, the ribosome will bind to the second cistron ribosomal binding site, that we can call simply as RBS. Okay, in contrast means opposite. So if the most bacterial mRNAs are polycistronic, means most of the eukaryotic mRNAs are monocistronic, so which can only code for one polypeptide chain. Okay, so in eukaryotes we don't want a specific ribosomal binding sites, because if you see eukaryotic mRNAs, there is a 5' capping and as well as 3' polyadenylyl tile. Okay, so at a 5' side there is a methyl group which is present in the eukaryotic mRNAs. So at a 3' side there is a polyadenylyl chain. Okay, which useful for the protection. Okay, because the eukaryotic mRNAs should, after transcription, after the post-transcriptional modification, it should travel from the nuclear pore to the cytosol. From nucleus, it will reach to the cytosol through nuclear pores. As well as there are a lot of enzymes that called nucleases which are present in the cytosol, which usually degrade the unnecessary nucleotide sequences. So we have to say this is our mRNA. So for that only the 5' capping and 3' poly A tail will be useful, and in translation the 5' capping is useful for the ribosomal binding site, and the poly A tail also having significance in the translation. Okay, sometimes a single mRNA will give multiple polypeptide chain in eukaryotes using alternative splicing mechanism. See, there is no separate cistrons. Using single cistron and using the alternative splicing mechanism, eukaryotic mRNAs will give multiple polypeptide chains. So that we will see multiple polypeptide chains means that are not different polypeptide chains. Same polypeptide chains may be with the loss of or with the lack of some amino acid sequence. Okay, so that we will see example. But prokaryotes lack introns. Why? It is only possible, that means with a single cistron or single mRNA, why only eukaryotes synthesizing multiple polypeptide chains? Because eukaryotes having introns and exons in the mRNA, but prokaryotes lack the introns. So prokaryotes doesn't have any introns. So introns means the non-coding sequences which are present in between or which lie between the exons. Here the exons are the coding sequences. If you see prokaryotic, that central dogma of molecular biology process, the transcription and translation will takes place simultaneously. But if you see eukaryotes, so after the transcription we will call it as heteronuclear RNA, hnRNA. So that heteronuclear RNA will undergo splicing mechanism, 5' capping, 3' polyadenylyl tail. So during the splicing mechanism, so the the enzymes or other proteins will usually remove the introns which are present in between the exons. So here the exons are the coding parts and the introns are the non-coding sequences. See, within the gene also we have a lot of non-coding sequences that we have already discussed. 98.5% of human DNA contain the non-coding sequences only. So not only in between the genes, within the genes also there are some non-coding sequences, but there is a specific significance for introns also. So you can watch those classes in Unacademy learning app. So you can search Maheshwai and you can watch those classes for free. Okay, so prokaryotes lack introns. So one cistron will give only single type of polypeptide chain. Okay, but some single mRNAs in eukaryotes will give multiple polypeptide chains. Okay, some introns in eukaryotes are much longer. So introns also longer in eukaryotes, not only the genome. So the longest known human intron is 17,106 base pairs. See how much long it is. See, that's why we have 98.5% of non-coding sequence, and which is lies within the titin gene. So titin is a gene which encodes a protein in the muscle cells. So within this titin gene we can find out the largest known human intron, that is with 17,106 base pairs. Okay, in comparison most human exons contain only 50 to 200 base pairs. See, the exon sequence is very less, it's a 50 to 200 base pairs only, but the introns are very long. So that is evolution. Everything will have a significance. Without that it won't be takes place. So everything will have some significance. Every organism, the life which is formed on the earth, the life which is evolved on the earth, only with the purpose. Without a purpose anything is not there. Okay, the typical human gene encoding an average sized protein is about 50,000 BP long. If you see generally the human gene which encode a mRNA, a protein which contain 50,000 base pairs. Okay, but within that 50,000 base pairs also, not only saying 50,000 because it's general thing. some genes may having more than 50,000 base pairs, but in that 95% are the introns only, and as well as the flanking non-coding sequences. Flanking non-coding sequences means the sequences which are present in the at the 5' side and the 3' side. Okay, so these sequences also including in the non-coding sequences only, those are usually called, that means the sequences which are present at the 5' and 3' end are called flanking sequences. So most of the 95% of genes contain these kind of sequences like non-coding sequences and flanking sequences. Okay, the fibronectin gene contains multiple copies of 5 types of exons. So it contains multiple copies. So if you see, there are 5 exons in the fibronectin. Each exon having multiple copies, same only, it's a duplicated one. Okay, so that we will see how they arised. Such genes evolved by tandem duplication of the DNA encoding the repeated exon. So we will see the tandem duplication, how and why it is happening. Okay, so which is probably by unequal crossing over during meiosis. Usually when meiosis will takes place, the meiosis will takes place during the formation of gametes like egg and sperm. During at that time only the crossing over will takes place. Okay, during crossing over what will happen, for example, take myself as an example. So I'm having half of the genome from my mother, half of the genome from my father. Okay, so when I'm producing gametes, that means when my body producing gametes, the father and mother, the diploid chromosome will cross over each other and exchange its genes. So after exchanging, what will happen? The haploid chromosomes only will enter into the gametes. So sometimes see, if you see some of the gametes, the haploid genomes contain a hybrid of both mother and father genes because of this crossing over. Okay, so this will usually occur during meiosis, but the crossing over should takes place equally. Unequal crossing over will leads to the duplication. See, that unequal crossing over will not always generate duplicated ones, it can kill organism also. For example, if that unequally crossed over gamete, if it is entered into the opposite sexual gamete, that is if it enter into the egg, automatically what will happen, some mutations, already it's a duplicated one, it is also a mutated one. So sometimes that may be advantage, so that may be selected by the natural selection, but sometimes that will not be selected by natural selection and it leads to one genetic disorder. Okay, so you can see here, so this is the homologous chromosome. Okay, so usually if you see this orange pink color and some blue color, there are 2 types of chromosomes here you can see. Okay, so it's a duplicated one actually. So if you see normally this parental, this for example, if we take this is a chromosome, these are the chromosomes which are under the meiotic process from a person X. Okay, so this person, usually this person X having this one part and this one part, that means blue color as well as orange pink color. Okay, so during meiotic process they will separate each other and they will form duplicates. So see how the crossing over is taking place between the non-sister chromatids. These 2 are the sister chromatids and these 2 are the non-sister chromatids. So the crossing over will takes place between the non-sister chromatids. So here it is equally happening. Why? Because the chromosomes lie each other or arranged each other during the crossing over process. Okay, so nothing issue, so it can form perfectly, so like this. But what will happen if the unequal crossing over occur? So that we will see. Okay, so you can see this here. So you can see this exon duplication and gene duplication, 2 things we are seeing. Exon duplication means only the exons will duplicate, there is a duplicated copy of exon will form. But if you see gene duplication, entire gene copy, extra copy will come. So how it will happen? See, the same thing what you remember, the same thing what you see in the last picture, and see there are two parental chromosomes. Instead of lying each other perfectly, they lied each other unequally. So the crossing over usually here it is taking place between the L1. So here the L1 is an interspersed element which is a mobile DNA actually. Okay. So see, for example, the mobile DNAs have the capability to move anywhere. That is also an important thing. Okay. So if you see, the crossing over should takes place equally, but these two chromosomes, parental chromosomes, are unequally placed each other or unequally lied each other. So the crossing over taking place here. Instead of taking place here, to these in between to these sequences, it's taking place between here. So automatically what will happen? See that recombinants. So one gamete will get, that means we can say simply one chromosome will get extra copy of exon. Here the duplication happened. This duplication also occur in the replication also. Sometimes sometimes the enzymes will do double replication, not for all the sequences, for some sequences. That leads to generation of another duplicated sequence. So another gamete which lack the exon 3. Okay. For example, if this one is selected, the first one is selected by natural selection, it will get the evolutionary advantage. So if it is not selected, the organism will die or it may contain any other disease. So if you see the second one, so it cannot produce a full polypeptide chain. If a protein is not produced, that leads to a disease or disorder, whatever. Okay, so that is, every protein is important in our body. If we lack a single protein also, merely it's to disorders or diseases that we will hear very commonly. See the gene duplication. See here the unequal crossing over taking place. Automatically one gamete will get the two genes and other gamete will lack the, other chromosome, because that parental chromosome will enter into other gamete. That's why I'm saying other gamete. So that another parental chromosome lack the beta globin gene, and it leads to the some disease. Okay, that organism will die. But if the first one, if it get advantage, okay, it can continue like this only. The evolutionary process happened. Not only this is one of the cause for the duplication of the genes. So there is another process called replication. There is another way, mobile DNA elements. It will copy and paste the sequence from one place to other place. So if you see a genome, it is having a wonders. We can't count what are how many wonders it is having. We can't count with the hand also, with our fingers. If we are searching something, it may link to some other more things. So this is about the exon duplications as well as monocistronic and polycistronic mRNAs. So let we see in the next class transcription units, simple and complex transcription units. So I'm gonna make short videos only. It may give more videos, more than 10 videos for each topic, but short videos may be helpful to you learn easily instead of doing hours of classes. And thank you. If you like this video, please like, share and subscribe to my channel. If you are having any query, if I am saying any wrong, you can put that thing in comments. So let me discuss that. Thank you so much.